Welcome to the Cold Spring Harbor Laboratory Organoid facility in Woodbury, New York. The development of organoid models is a tremendous advancement in paving the way toward personalized medicine for pancreatic cancer. Today, we take you on a tour of the state-of-the-art facility where you'll see firsthand how researchers use organoids to match the best therapy to each patient.
The idea of modeling a patient's cancer in the lab has been an exciting idea for a very long time, and for a long time all that we really had available was something called 2D cells, where you took a dish and you had 2D cells that would adhere and attach to that dish. Those cells in a lot of cases don't really fully represent the patient, and so they're not necessarily the best model system. So in about 2010 or so, Hans Cleaver's lab actually made huge advances and really developed something called an organoid model. And that organoid model actually allowed you to grow a 3D structure and grow it in a matrix in the lab. And that actually really recreated how the patient's tumor would respond a lot better than 2D cells stuck on hard plastic. It mimics the human body kind of in this squishy environment. So a few years after that, Dr. Tuvison actually worked with the Cleaver's lab to develop the idea of pancreatic cancer organoids. And the Lustgarten Foundation was really instrumental in actually funding and supporting that early work to develop these pancreatic cancer organoid models. And it's been about a decade since then, and we've made huge advances in actually developing patient-derived organoid models. We've now had over 1,000 tissue samples that we've received and made hundreds of organoid models. And it's really been the Lustgarten Foundation that's supported that and driven that.
Our first stop on this tour is the lab refrigerator that houses a pink substance called the media. The media contains several dozen different growth factors and agents that are needed for organoids to grow. Next to the refrigerator in the lab, you'll see the incubators. These incubators are temperature, humidity, and CO2 level controlled to create the optimal environment for the growth of organoids. Inside one of these incubators, you'll see stacks of trays containing organoid cultures in plastic dishes. The organoids grow inside those small, clear domes in each dish that mimic the consistency of the human body. Let's take a look at one of these organoids under the microscope with Amber Habowski, a postdoctoral fellow at the Tuvison Lab in Cold Spring Harbor, New York.
This is actually that dome that we talked about. So that's that dome you can see with your naked eye. If Luce jiggles the plate a little bit, you'll actually see there's a little jiggle to it, because this is like a squishy substance. And this is actually, so this is in 3D, and you can kind of go up and down through the planes to see the 3D-ness. But each of these round spheres is a 3D organoid. This is a very cystic organoid, so the center is hollow. Sometimes they'll be more filled in, and sometimes they're smaller. This is like what we'd say a very like tumory looking organoid, where it's very dense and thick and the cells are kind of overlapping on top of each other. So Luce will keep growing and expanding this line. So what she'll do is she'll let this grow maybe a little bit longer, and then when it's ready, she'll do something called passaging, where she'll dissociate the cells, break all of these organoids down into single cells, or close to, and then they'll actually grow back up and form organoids again. And then once she has this kind of single cell suspension and we have enough cells, we'll use some of our fancy machines over here to actually plate those cells onto a plate. Traditional drug screening use something called the 96 well plate, and this is really large well size. And so in each of these wells you'd put quite a few cells that would make organoids and different drugs. But we really wanted to try to pack in more drugs and more different dosages of the drugs. And so we started to miniaturize things. So you could then go to a 3D4 well plate, which looks like this. So you can see much smaller well size. So you could have, on one plate, you could have more drugs you're testing. And then we miniaturized further by going to a 1536 well plate. And this is what we do for most of our standard screening now. So you can imagine on this, we can fit, 50 plus drugs in triplicates with dose response curves and really get a lot of data packed out of one plate. The challenge with this is you can't physically pipette into this, right? So on this, you could manually do it, but on this you can't. So we actually need to rely on our automatic fluidic systems in order to put the cells and dispense the cells and the drugs into here.
This machine automatically dispenses cells in the plate, allowing for a quick, efficient, and sterile plating process. Once plated, the cells are brought over to the acoustic liquid handler, where tiny droplets of the drug being screened are added to the well plate of cells.
And so what we actually do is we have something called a source plate that has the stock drug. This plate goes in and flips upside down above it, and then this machine uses acoustic sound waves in a little water bubble and sends little pings to the source plate that then send little tiny droplets of the drug to this plate. And then we flip it over and then add the cells.
Next stop in the drug screening process is the plate reader. The Organoid facility researchers add a chemical that gives off strong light signals when the cells are alive and low light signals when the cells are dead. Those high and low signals are color-coded and displayed on a computer. The red areas reflect live cells and the blue and purple areas reflect dead cells. By looking at the colors, the researchers can tell which drugs are working and which are not. Once the organoids are drug screened, their data is stored in a software program developed by the team at the Organoid facility. Researchers can go in this software and look at any given drug relative to the rest of the biobank.
So what pops up is this is what we call a violin plot or a dot plot, and each dot on here is an individual organoid sample that's been drug screened. This particular one is a recent organoid from our Paso 1 trial, and you can see this is that organoid here in orange. And usually what we say, so this is based off of the AUC, which is the area under the curve. So this, from the drug screening data that we have these different concentrations of drug, and then we look to see how viable or how alive the cells are. So as the concentration increases, the viability of the cell will go down. For some drugs it goes down really quickly, for other drugs it goes down slower, some drugs no effect at all. So then once we have these dose response curves drawn, we can calculate the area under this curve or under this dose response, and then we can use that to compare across the organoids and across the biobank. So for this particular one, the other plot we were looking at 5FU, you can see here's that line in blue, and then that alone doesn't necessarily tell us very much, right? But when we compare it across a few hundred patients, then we can actually look and see for this particular organoid, this is a more sensitive. The dose response curve was more or less shifted, and this would put it in the sensitive range, whereas these organoids up here would be considered more resistant to 5FU.
It is helpful to build out a biobank like this because some drugs require a lot of the drug to be effective. It isn't always known if a drug is effective in a patient versus an organoid, so it's important to look across a whole cohort, as opposed to comparing a dose response for different drugs.
If we know historically about a third of patients might respond, we can kind of say, okay, maybe a third of our organoids should be sensitive to a given chemo. One of the things we can do is we can look and say, okay, which chemos, right? If we know it's about a third, we can look at all of our chemos and say, is there anything that they are sensitive to? And then when we find some organoids, which we do find, that are just pan-resistant, resistant to all of the chemos, we actually then go and look through all our targeted therapies. And we say, is there something else that they're sensitive to? And sometimes we find something, but there's some times where we don't find anything. And on that one, that's where we need to develop more therapies. We need to try more experimental drugs and find something else that might work. Immunotherapy, we can't really test very well in our system right now because we're focused mostly on just growing malignant cells. In the future, if we have immune cells and more of the microenvironment cells, we might be able to test some of these other therapies that actually influence the microenvironment. But for now, we're limited to things that are just targeting the malignant cells.
Let's head over to the Droplet Digital PCR Machine. This machine is used to look for KRAS mutations. Researchers take a sample of an organoid, make little droplets from an individual DNA sample, and run a PCR reaction through each of the droplets. Next, the DNA template is compared to primers that are very specific to the KRAS mutation. If the primers and DNA template match, many DNA copies are made. If they don't match, no DNA copies are made.
We tag a little signal or color onto those copies. So then when the copy is made, we get a signal. So you can actually see there's this channel 1 amplitude and channel 2. So when we have the amplitude, you can see we actually get a signal versus a different type of signal. And so if we look at one sample in particular, so this is looking at one sample, and we know this is the KRAS mutation signal. and then this is the KRAS wild type signal. So in this case, we see this cluster here. So we know that our sample we're looking at has a KRAS mutation. And this allows us to confirm that the organoids are cancerous. They have a KRAS mutation, and then we can also compare, this is the KRAS wild type signal. We can compare how many copy numbers there are, if there's an amplification, if there's, a certain ratio of wild type to the mutation as well.
Our last stop on the Organoid facility tour is the freezer room. Here, you'll find different freezers at various temperatures storing DNA, RNA, tissue pellets, and other materials that researchers biobank and store for future use. The cryostorage system uses liquid nitrogen at -170 degrees Celsius to store boxes of frozen, preserved cells that last for decades.
Thank you guys so much for taking the time to come visit the Organoid facility here at Cold Spring Harbor Lab. We are immensely appreciative of all the support from the Lustgarten Foundation and donors like you. We'd also like to give a huge thank you to all the patients that make this research possible and are willing to donate their tissue so we can further advance pancreatic cancer and our understanding and hope for a cure in the future. Thank you.